I'm Wendy Hartsock, science and peptide enthusiast. In this episode of Exploration Science, I met up with Cameron Pye and Joshua Schwochert, the co-founders of Unnatural Products. They discussed the path to starting a company while finishing their PhDs, engineered serendipity, and the power of macrocycles to engage intracellular targets. Thanks for agreeing to do Exploration Science.
Yeah, definitely. Thanks for having us.
So first off, I wanted to kind of get to know more about your backgrounds and how you got to where you are, and also how you know each other. So Cameron, you happen to be on my speaker view right now, so I'll start with you.
Sounds great. Yeah, thanks, Wendy. Josh and I have known each other, I think, for close to 11 years at this point. So we met each other actually on our grad student visit to UCSC when we were still in undergrad or shortly thereafter for both of us. So yeah, we've been working together in one context or another for quite a long time. My background, I went to a small liberal arts college down in Southern California called Whittier College, but from Georgia originally. Came out west looking to do some science. Almost lost me from chemistry to philosophy for a while, but luckily saw the light and stuck with it. And yeah, the rest is history. Came up to UCSC, joined Scott Loki's lab to do my PhD, thinking I was going to do more organic synthesis, but really fell in love with Scott's chemical biology lab and the work that was going on there in the macrocyclic peptide field. And then he kind of identified some of my computational chops and really wound up drifting heavily into chemoinformatics and more theory work with Scott by the end of my days there. But yeah, Josh, you want to chime in?
I also went to a small liberal arts college in Southern California, though it was Concordia University. And it was interesting because that school didn't really have, there was no graduate program, so there wasn't a whole lot of active research. But I was initially planning on going to medical school. I had volunteered in an ER in high school for a few years. That was kind of what my career trajectory was. But one of my organic chemistry professors saw that I was really engaged in more of the chemistry kind of research side of things that we talked about in class. And so he set me up with an NSF REU up at Washington State University. And I got my first exposure to graduate level chemistry and, actual kind of proper research in a lab. And I fell in love with it immediately, decided I wasn't going to take the MCAT and instead take the GRE. And then from there, really had no idea what I wanted to do because I loved biology. I was a pre-med major, but also had a chemistry major. And I also was excited about chemistry. And when I interviewed at a couple different places, Santa Cruz really had, I think, a good energy for me. And one of the things that really got me excited was there was really good RNA structural biology. So this was kind of at the, oh my gosh, we might be able to solve the spliceosome the way we solve the ribosome. And there's a couple really good researchers at UCSC. So I came to UCSC expecting to do structural biology and then rotated in Scott's lab. I was in an umbrella program where I could kind of pick and choose different labs. And on a whim, on my final rotation, Scott convinced me to work in his lab. And I really just fell in love with the pretty clear track of coming up with a hypothesis, making molecules, and then testing them in a biological system. That kind of chemical biology synergy of we want to modulate biology, but to do it, we have to understand chemistry. And you can directly make tools that are applicable. That was really cool. And I think it brought together the love of chemistry I had from kind of a synthetic perspective, but then also really the biology is big, hairy, and complex, and we need all the tools we can get to understand how it works. So I ended up doing a lot of natural product total synthesis, easy total synthesis in the sense that they were cyclic peptides. So not a whole lot of stereo centers I had to make myself. But yeah, started working with Cam. Him bringing computational approaches into Scott's lab, which was relatively new expertise to have in the lab, really let us collaborate a lot on a bunch of different projects and let us ask some cool questions, both from a modeling perspective, but then also a chemoinformatics and parallel data processing perspective. So we
worked my way onto a bunch of papers without doing much actual lab work too. So that was a win-win for me. But yeah, and then towards the end of our PhDs, I think Scott's lab was always really industry gazing. We did pretty much all of the work was funded through pre-competitive pharma collaborations at the time. And so, we went to kind of industry focused conferences. We were asking these pharmacokinetic questions, which were often more kind of industry and medicinal chemistry oriented than typical academic groups focused on. And so I think we just kind of knew what startups in the macrocyclic peptide space looked like, and kind of what pharma was up to, and had a bit of an aha moment about 2/3 of the way through our PhD about kind of the role of cell permeability in these macrocyclic peptides and our ability to kind of predict where that might be. And I knew Josh was going to go off and do amazing things in academia if I didn't quickly sequester him to the private sector. So founded the company towards the end of our PhD work, and then wound up graduating and jumping into it full hog. So that's, I think, going on 3.5 years ago at this point. But
I have several questions, but one is, Josh, I'm curious, did you ever go back to that organic chemistry professor and tell them, where you've ended up? And
actually, I reached out to him immediately after graduating and getting my PhD. When we were in the garage days of the company, it was a little bit like, tell people, but we're not quite over that initial hump yet. So maybe 1.5 2 years ago, I reached back out and I went and gave a seminar to a bunch of undergraduates at my institution and kind of talked to them about my career path. And for me, I didn't really even appreciate that getting a PhD and being a researcher was really a path. I also went to kind of a small private high school, and there wasn't really a big STEM focus there. And so, just being told, hey, you can go study science. They're not just like people from a few hundred years ago who, were playing with prisms in the basement of a castle. Like, there's really a big active science community.
Yeah.
That was really important for me. So
that's great. I keep meaning to do that with many of my undergrad or high school teachers, to be like, hey, I actually did do something, like I'm not in prison,
Totally. Yeah. I made it through.
Yeah. So tell me about starting a company. Did you actually start it during your PhD? It sounds like you actually started the process while you were finishing your PhDs.
Yeah, I incorporated the company at least when we were just wrapping up, I think. But truth be told, incorporation is kind of like a legal mechanism, but I think if you have a name and start mentally assigning your ideas to a company name versus yourself, then you've kind of started a company. And the rest is about conviction and operation from there. But it was maybe 6 or 9 months before we graduated. We kind of had the very rough idea, I think, of what we wanted to do. Like I said, we're kind of armed with some new data sets and new know-how, and we're trying to figure out how to translate that into a viable commercial application, which we knew was there. So founded the company and kind of told ourselves this is what we were going to do next. We disclosed kind of all kind of current inventions to UCSC. But wound up not really needing to take a license on kind of what we were thinking about originally. And so then kind of saw the opportunity to graduate and not have to kind of come up with initial cash for licensing from the university. So our thesis committees kindly let us really do an accelerated thesis writing and proofing thereof, or lack of proofing, I think in my case. So I think Josh and I each wrote our theses in about 2 weeks, and we graduated on the last day of the quarter that we could before we were going to have to get reset by another 3 months.
Back to back, actually. So Cam gave his dissertation, I drank a glass of champagne, and then I walked behind the lectern and gave mine, which was good. So we had a pretty big Venn diagram overlap of friends and family. So it made it easy for people too.
It made it easy for us. I felt bad for all of our friends that had to sit through 2 thesis dissertations back.
And were the committees the same too?
No, not quite. Though there
Yeah.
is some overlap there as well. But yeah, I had a committee member was not too happy with my accelerated thesis writing approach, and I was holding his signature hostage on me. And like the day I needed his signature, he was like AWOL, and I was tracking him down through campus until I finally found him, and he gave me a start talking to, and then signed on the dotted line. But
yeah, I think in the earliest days it was we just stole away some time to think about the company and what we were going to do next. And I think in the same way, some PIs will be nice and let late graduate students spend time applying to jobs and maybe doing internships. Scott gave us leeway to, spend some hours on the company. And we found an abandoned office in the chemistry building that we set up as our little temporary office and all that. So it was kind of a relatively smooth transition where, although there was a rapid exit, we were kind of simultaneously wrapping up our grad work and kind of thinking about what it would look like to start a company next.
And what does Unnatural Products look like? What do you guys do? What do you offer? What are your collaborations?
Yeah, definitely. So, it's really the extension, I think, of some of that work we were doing in Scott's lab, which is, Unnatural Products' goal is to translate kind of the properties of finding natural products, macrocyclic peptides and macrocycles into synthetic systems for the goal of drugging previously undruggable targets. So really expanding the scope of small molecules to protein-protein interactions inside the cell, using this macrocyclic peptide scaffold as kind of the general solution modality to that problem. And yeah, I think, kind of as Josh alluded to, I think our two personal skill sets kind of typify what the underlying technical thesis is too, really a marriage of computational approaches and empirical wet lab science approaches. So, we bring some machine learning component to the property prediction. So really being able to do, QSPR style modeling wasn't really possible because we were really data constrained previously in the space. And that was some of the work we did during our PhDs, but also a ton of kind of in silico infrastructure that lets us carry out the wet lab operations and the scale and efficiency that we do, which is, kind of Josh's domain. Where we kind of can synthesize hundreds of thousands of compounds in milligram scale directed parallel chemistry, and then more recently have booted up, more combi approaches for de novo or focused library generation in the case of like affinity selection mass spec approaches, and then more recently phage display and mRNA display coming soon. So really kind of a suite of discovery technologies from trillions of macrocycles to singleton macrocycles, kind of encapsulated and guided by the in silico modeling to let us drive from biological hypothesis to drug candidate.
That must be nice to have that all in-house as you're figuring out the data sets that you need. I was just speaking with Ava Liss and Yushan Lin about, what does it take, how many sequences do you need? And of course, that's an unanswerable question, but it must be nice to start learning, like, for a particular project, how much you're going to need to put into it, and then to have the testing in house. That's got to feel pretty good and really streamline the process.
Yeah, it's a recent addition for some of those technologies I just mentioned. So
yeah,
just broke the beer bottle over the bow of some of those technologies.
Maybe not streamlined yet.
but rapidly becoming so, and already starting to see really the power of having all of that. I think in our hands. So,
but it's great to have the experimental component in house. We do have collaborations and we do some external work, and their turnaround's just always longer. And, even if they're a really great partner, it's different when your team is doing the work and there's kind of a different level of emotional investment there and excitement and all that. And so being able to actually make the compounds run the experiments in house, I think is really satisfying. And when you're trying to predict properties for things, you're wrong a lot. So being able to run a lot of experiments internally is a great way to, quickly find the successes and build off them. And being able to pair computation with parallel experimentation, you also can be wrong in a good way, where you find things you weren't expecting. And so if you're throwing a lot of experiments at the wall, you can find things that kind of change your understanding, and then leverage that and learn from that and move on. So I think that's really been useful to just be able to push a lot of experiments forward and then use that to kind of build your data sets, build your understanding on the fly, as opposed to trying to look at what's at the public domain and come to some final conclusion about what the best molecules are and then go and test that one thing. It's really more of a running experimentation, running modeling approach.
Yeah. What are some of the most surprising things that you've found?
Oh, that's a good question. I don't know. I mean, there's like, the medchem stories are always entertaining to the medicinal chemistry crowd, but maybe not beyond that. But yeah, we had some interesting things. We really take like a data first approach. It's this kind of ligand based design platform is the medicinal chemistry engine. So we kind of feed it with a hit from one of these more parallel approaches, and then kind of do this iterative focused library generation. And sometimes that's guided by structural biology. Like if we have a co-crystal structure, we'll impose that knowledge on the system. But on one of our lead programs, actually really proof of principle work, we were doing like an MDM2 P53 series of macrocycles, and they were kind of pseudo C2 symmetric. There was kind of like a series of hydrophobes on one side, a series of hydrophobes on the other, and some kind of polar stuff in between. And at one point, we didn't even realize it, but we had like a binding mode inversion, like the molecule had flipped, and it was kind of one of those things where, yeah, if you're just kind of letting the successful molecules dictate the generation of the next successful molecules, you might not, notice that. But if you're really tied to a rational design hypothesis, you would kind of never let that happen. And so I think there's moments of serendipity like that where we kind of, engineered serendipity, I think, where we kind of cast the net in the right direction, but don't try to, impose more knowledge on the system that I think we're willing to convince ourselves that we have, and we see a lot of kind of fortuitous kind of binding modes and properties kind of emerge out of that kind of ecosystem that I think if you take a really kind of traditional small molecule medchem approach to the problem, you just never see. So yeah, I don't know. I think that's one.
Yeah. How about you, Josh? Anything that sticks out? You're like, whoa.
I don't know. Actually, we recently got co-crystals of some of our potent inhibitors against their protein targets. And that was really cool. I think I'd spent a lot of time, academically in Scott's lab. Our focus was really about how do we optimize properties, and binders were few and far in between. It was much more a pharmacokinetics ADME exercise. So the idea of getting crystal structures of, nanomolar or picomolar compounds that we had designed and synthesized against their target was something I was really looking forward to. And sure enough, when we did finally get some really high resolution structures, there was SAR that we didn't understand because we had only, some structural data from the very start of the project. Where we were at that point, every amino acid had changed. The binding mode had certainly moved around. And there was one position, it was a small little heterocyclothiazole, and the structures just really liked it. All the compounds were really potent with that modification there. And in our mental model, we thought that it was sticking out into space, but it turns out it's making a beautiful kind of pi stacking interaction with the planar amide bond of a peptide backbone kind of adjacent to the pocket. And there was another residue that we also thought was probably solvent exposed, and it turns out it had nestled underneath the side chain of another residue there. So there was all this SAR that we had a good understanding of. We knew that these amino acids were really favored at these positions, but our mental model didn't understand why they were so favored, and then actually seeing the atomic coordinates and being like, no, that's a really specific interaction, and it makes sense that that's the only thing that's tolerated there. That was really satisfying and pretty shocking. I had assumed they were just wiggling out into space and being kind of hydrogen bond acceptors out there to give us some water solubility, but they were making really specific contacts with the protein.
Oh. So talk to me about beyond the rule of 5 and some of the limitations that you've encountered. So, both those aspects.
Yeah, definitely. I think Bro 5, as it's sometimes called by its barrier proponents, is it's this kind of evolution, I think, of small molecule space. The extended rule of 5, I think, has existed as in the literature parlance for a little while. But yeah, at the end of the day, these are rules of thumb that are retrospective, looking back at the work that Pfizer had done on what were successful molecules and what had gotten FDA approved. But by no means were they prescriptive, right? And I think that beyond the rule of 5, particularly the molecular weight rule of 500 Daltons being kind of the cap of quote unquote drug likeness, we've known as a soft cap for a while. And I think, in synthetic small molecules, we've seen things creep well beyond that. We always like to point to navitoclax and venetoclax as some really heroic medchem work from AbbVie and Genentech's teams to get some really large small molecules to break up protein-protein interactions. But we've also known it's a rule of many exceptions thanks to the natural products that we've been using for decades before venetoclax, which threw caution to the wind. And I think for a long time, natural products occupied this special space in medicinal chemistry where it was like we can find those, but we can't make them. It was really like that we could do semi-synthesis or tweak these scaffolds that nature had engineered, but there was so much more kind of sterically and conformationally complex than a typical small molecule that kind of designing those de novo would be really challenging. And I think cyclic peptides are what we see as kind of a niche of that kind of complex chemical space, natural products like space, where we can kind of, grab it by the horns and actually be able to start to engineer design in that space. And that's thanks in no small part to their peptidic nature. I think we're focused on highly non-natural cyclic peptides because they kind of bridge the gap between really complex natural products and more easily synthesizable things, thanks to amide bond formation. But the landscape is more jagged. It's harder. The larger you get to balance property spaces and target engagement. Chief among those is cell permeability and aqueous solubility. Kind of form this razor's edge that you have to be able to balance while you're doing your medicinal chemistry to optimize these things to have those small molecule drug-like properties and still bind some target of interest. And unlike nature, we don't have a few billion years to evolve these things. We're on slightly more expeditious timescales in startup life, and so I think we've found the tool sets we've generated, these in silico and empirical techniques, combined with a little bit of our know-how and really the fantastic team, has been able to kind of do that reproducibly in the space. But it's still challenging, even for us. I think that we see this as really a blue ocean opportunity to kind of engineer Bro 5 compounds to behave like RO5 compounds. But we've always known it's possible, I guess. We've just known that it's harder than in the small molecule space. But the biological targets and opportunities there, I think, are too great not to, kind of try and crack the nut. So yeah, that was a long diatribe. But Josh, you have anything to add to it?
No, I think you kind of asked what are some of the challenges. There's our primary focus is disrupting protein-protein interactions, and so that a lot of traditional HTS and kind of screening strategies are enzymatically focused, right? So if you're looking at disrupting something, that's kind of a whole different assay set. So, and for a lot of these large compounds, you're able to drive really low potency or really high potency, and so there's kind of ongoing efforts in the community to continue to push the limit on those assays. Merck just published a great series of papers about their PCSK9 macrocyclic inhibitors, and they get these assays that can read out down into the low picomolar affinities with a competitive assay, so it's amenable to HTS. So I think the community is kind of building on new techniques to deal with the potencies, the types of interactions, the type of protein targets you're hitting. Boehringer Ingelheim just published a great paper looking at they're doing, it looks like internally, some cool natural product cyclic peptide work internally. And they ran into some limitations of traditional trans-wall permeability assays. And not like inventing a new assay, but just how do you deal with these larger lipophilic compounds with high protein binding, and how do you get a stronger in vitro in vivo correlation? So I think to some extent, the challenges are just that our assay set and the standard conditions and everything have kind of been built around rule of 5 compounds and rule of 5, ways to affect protein targets. And so now that we're moving into bigger, more complex compounds, but also bigger, more complex targets, the tools just have to change. And the thing you grab off the shelf isn't necessarily the same tools you would have if you're making another generation of kinase inhibitors.
Sure. Yeah. So I know, so you're targeting protein-protein interactions and you're balancing, I liked the word chameleonic compounds. You're really thinking about, getting into the cell and how do you balance those characteristics that allow for the lipophilicity, but also the solubility. As you're doing that and designing these molecules, are you also thinking about further downstream and how delivery will be affected by the lipophilic nature of these compounds?
Yeah, I mean, I think, at the end of the day, when we get our compounds to behave, they truly behave, like the cyclosporins of the world, which is to say big, greasy, small molecules. So, their oral or propensity for oral delivery is the same of that of a small molecule. You can freely diffuse across the gut. And then, you just need to survive first pass clearance to the liver and not get kind of fully excreted through hepatobiliary clearance, and then make your way into the blood and distribute into tissues like small molecule would. So I think, our goal is not to reinvent any wheels on delivery. It's really to, achieve the profiles of smaller molecules that we've come to know and love with these larger scaffolds capable of the picomolar binding of challenging intracellular PPIs that Josh was talking about. So I think, that being said, there are some, kind of formulation challenges with lipophilic molecules in general. For instance, like, IV dosing in preclinical rodent models is notoriously challenging because you need to have fairly small, short injection volumes and times. And if you're limited by aqueous solubility, then it really hampers the effective dose you can give. So there's, kind of weird operational quirks of that. They aren't new to this particular molecular class. I think they're shared of poorly soluble aqueous compounds as well. I'd love to bring up venetoclax, and apologies to the med chemists that worked on it, but it's listed as like sparingly soluble in water, and it's like down in the double-digit nanomolar and pure buffer or lower. So it's like, we know these are circumventable challenges, but they're challenges nonetheless. But yeah, I think for us, we don't think about these things, I think, like some of the other peptide delivery kind of strategies do, like whether that's like, particle encapsulation or, these cationic cargo tag kind of strategies, because we're really achieving small molecule like profiles. It's like, it's the devil we know. So I think that it's really all the lessons we've learned over, the past 100 years or more of small molecule, medicinal chemistry and lead optimization, and the way those in vitro to in vivo correlations work, we can kind of parlay into this molecular class as well.
Yeah. What do you think some of the biggest opportunities are for you? I mean, I know obviously if you can take over small molecule space and get into protein-protein interactions, it's kind of endless and vast. But what do you see as maybe opportunities that are the lowest hanging fruit? But then beyond that, what are the ones that you really want to go after?
Yeah, I mean, there's in kind of the biological target landscape, we think about that a lot these days because it's kind of the world's your ocean. Where do you set sail first? And it's like I think at the end of the day, we started with kind of the high hanging fruit in oncology, these quote unquote obvious targets that we've been working on for decades, things like P53, the RAS family, beta-catenin and MYC, I think are, what Greg Verdine calls the 4 horsemen of cancer, and BI calls, cancer's big 4. I think, and we're starting to see small molecules put chinks in that armor. I think that, like I said, it's just harder out here, and we think macrocycles are a good general solution to this problem of large, beyond rule of 5 small molecule med chem. But but there's other crafty strategies that we've seen, like in the RAS space more recently, covalent strategies, or even just kind of bidentate strategies in the case of Mirati. And so I think, there's going to be other players in that space as well. I think Fog just announced the IND on their beta-catenin stapled alpha helix. So I think, there's other folks there, but it's a huge opportunity space still. RAS is a big complex family of proteins, and not all give way to the G12C strategy. And so I think that's kind of where we hung our hat as, I think, the lower branches, the high hanging fruit. And some other folks are starting to reach up there as well. I think you can also look at, this huge swath of kind of untapped biology and the transcription factors, the dubs, there's this kind of suite of non-classically druggable targets that we can start to look at. But it's interesting, you run into this kind of target validation problem pretty quickly there, where we don't even have tool compounds against all of these things. So just kind of the early literature around some of the nascent biology that we can start to go after, it's a lot less validated. It's a lot less kind of investigated as to what perturbing that does in the system. And I think modern CRISPR knockout series, the Broad step map, and that can kind of shine some light on that and point you in the right direction. But I do think that that's kind of the high hanging branches with some juicy fruit on it, but some of them might be a little spindly. And I think we're going to kind of have a balance, I think, of risk in our pipeline as we move forward here. But Josh, you've been probably thinking about this more than I have lately. So
no, I think what Cam hit on, that there's been a real increase in the sophistication of the bioinformatic approaches to find new targets, where it's not just looking at cells in culture and their dependency on certain genes, but it's correlating it to biomarkers that would identify patient populations and outcomes of patients that harbor this specific type of dysregulation. So I think that's kind of a second gold rush. I think it's the functional version of mining the human genome, where now we don't just know the proteins, we have some sense of their role in disease, or at least their role in driving specific types of cancers. And so when you're faced with this kind of ever-growing list of targets that are validated from these omics approaches, that's where, if you have the traditional tool set, you really have to try and identify the enzymes. But if you have these other new modalities, whether it's our approach of hitting protein-protein interactions, or PROTACs, or any of these kind of emerging beyond rule of 5 chemical strategies, you can look at the whole group of those targets and not just the ones that are compatible with traditional chemistry. So I think that's really the exciting next set of problems we're going to start tackling. And like Cam said, the challenges there, and why that fruit's even higher, is they're new targets without a lot of validation. You have to do the target validation in real time. And we've run into that internally, where, if you don't have any tool, if you're trying to make the first inhibitor, there's no tool compounds to validate assays and everything. So there is a little bit of you have to pave the road as you go down it, when you're working on kind of this newer biology that doesn't have 10, 20, 30 years of big pharma effort against it.
Yeah. I have another question that maybe it might be stupid, but I'm going to ask it anyway. So usually we talk about, disrupting protein-protein interactions, but you brought up PROTAC, where you're, bringing two large molecules together. And of course that's to degrade it. But have you also looked at, and what made me think of this was when Cam started talking about transcription factors, doing multi-molecule engagement to turn on something rather than to just inhibit.
We've definitely thought about it. We haven't put it into practice yet. I think maybe one to talk about some work other folks have done, Warp Drive, out of Boston, and now their compounds are being developed by Revolution Medicines. They had a strategy about trying to find molecules that brought together two disparate proteins. In their case, they were working off of these natural products that bind the cyclophilins, and kind of modulating the other half, and just using cyclophilin as a really big orthosteric block, kind of no matter where you're binding on the protein. I think as we refine de novo discovery strategies, it's a little more nuanced when you're trying to make a molecule that has binding to two different interfaces, and you kind of have to think about your initial assay and screening workflow differently. I think it's on the horizon, but it's one step further. You're doing medicinal chemistry, not just against one target, but you're doing it against two different targets. And I think that's one reason PROTACs, the first wave of PROTACs were these bivalent degraders where you kind of had a small molecular weight ligand, you had another small molecular ligand, and you put something kind of flexible between. And the next generation of PROTACs are these kind of monovalent degraders where you're doing something more like a natural product canonically would do if it was bringing two proteins together, where you bind one protein and create a new novel interface that now has complementarity to the other protein. So I think in the larger molecule, more challenging target space, that's kind of on the horizon, but not active.
I think it all started with cyclosporine after all, the original molecular glue, and it's FK506 and its brethren, I think, really kind of, I think, partially inspired the field that this is kind of possible in the first place, that a smaller molecule can kind of form these ternary complexes. So I think, and there's kind of this hidden conceit in the PROTAC kind of, world that every molecule or every protein target they'd like to degrade has some kind of small molecule binding site that you can generate either a monovalent degrader or bivalent degrader against. But we know that some of these things are really Teflon to kind of HTS screening approaches, and so I think larger molecules like macrocycles are kind of poised to potentially really expand the degrader kind of strategy space in ways that I think small molecules are going to have kind of issues, just like finding small molecule traditional inhibitors of those. So I think there'll definitely be cases where we find the cryptic pockets and the degrader strategies kind of take what used to be an undruggable protein interaction and turn it into a druggable strategy. It's also a fundamentally different MOA for degrading that I think it remains to be seen what that plays out like fully in the clinic, but some of the early data out there looks inspiring. So I'm super excited about the field in general, and imagine that we'll dip our toes into it here pretty soon, and have some ideas that are currently on the whiteboard, but hopefully reduce practice here on how to kind of leverage that in the macrocycle space. But yeah, I think there's a lot of interesting opportunities, whether it's degrading or just bringing things together to turn on or off function. I think is definitely kind of a nascent field. But
I think KRAS is a good example, because KRAS is one of those targets that's traditionally just been unligandable or very challenging to ligand. And so, great work out of Kayvon's lab found that, you could give yourself some affinity by making a covalent interaction and use that to find really weak binders. And eventually the compounds that were at Wellspring and then kind of inspired Mirati, Amgen, and everyone else, finally there's some traction there. And then Mirati's got some non-covalent inhibitors in the G12D space, but they're all roughly the same chemical vein, right? And just within the last year or so, there have been a handful of publications with cyclic peptides with totally different binding modes, all binding into that same pocket. So I think the argument that the additional molecular weight can give you varied binding modes, affinity in places where it might take decades to get traction with a small molecule, it's definitely true, because there's a novel series all the time against these very challenging targets identified from these large selection techniques like mRNA and phage display. So I think the concept of often undruggable targets are unligandable, and that larger molecules can solve that problem, I think that continues to bear out, that really the benefit of these larger compounds is to bind interfaces that are just challenging to get good affinity when you've got just less molecular weight, less surface area to play with.
As you're out talking to folks, whether it's fundraising or at conferences or forming collaborations, are you finding that there's more acceptance of the peptide space, large molecules? I did my PhD and I finished in 2010, and it was in a med chem department, and there was a lot of peptide hate going on, and so I'm curious to know, as the field has progressed so much, what do you encounter when you talk to folks?
Yeah, I mean, it depends on who you talk to, obviously, but I definitely think there's kind of another wave of resurgence of interest in peptides in the broader sense. I think, we sometimes refer to our macrocycles as amide-rich rings as opposed to so peptides to avoid some of the scorn that you mentioned. But I think there's kind of the traditional peptides were often, these kind of like endogenous hormone mimetics, like in the GPT space and GLP-1 agonists, and insulin and melanocortin systems and those kind of things. There's been incredible successes there, I think, in pushing the quote unquote drug likeness of those, but mostly through kind of like improving on peptides, kind of natural state. In terms of moving towards these like getting peptides to behave more and more like small molecules, I think we're also starting to see interest there. And then the cyclic peptide community, I think, is one of those that has made inroads in a lot of, med chem and modality teams inside of pharma. I think the PCSK9 story coming out of Merck recently, with their oral PCSK9 macrocycle, and Novartis published some work not too far behind it, is kind of shows that kind of you can kind of get halfway there right now, and there's interest. We can get oral delivery against extracellular targets in the kind of combined efforts of a lot of smart folks at pharma and quite a bit of effort. I think we're going to start seeing more and more successes there. And there's this kind of growing peptide drug conjugate field, which I think is also gaining traction. So I think we're starting to see some creep into, I think, acceptance in kind of where these things are preferential to antibodies from either route of administration or like manufacture and control over synthesis kind of perspective in the conjugate case. And I think what hopefully we'll be able to kind of get everyone to drink the Kool-Aid on these things can really be drug-like once we start kind of delivering more and more truly small molecule like peptide scaffolds. And we're not the only ones doing this, right? Like I think, the stapled alpha helix crowd has been, pushing the limits on that modality or submodality as well. And like I just mentioned, Fog Pharma, I think, has some probably very compelling data that they'll be revealing on their beta-catenin scaffolds. I think those things aren't quite small molecule-like quite there, but it just shows kind of, again, kind of more and more kind of conviction. And I think what the greater peptide modality, if you use peptide in the broader sense, can do. And I think we've really just kind of scratched the surface. I think that we're going to start to see more and more of those kind of success stories, both coming out of pharma, biotech, academia, and everywhere in between. So, yeah.
So I usually like to ask, you kind of segued into this, of what you're most hopeful for the next 5 years, like maybe, what you see as coming out. And it could be for Unnatural Products or just in general in the field. So yeah, just to pose that question, what are you most hopeful for in the next 5 years and excited for?
Yeah, I think the comments I made earlier about there's a lot of great targets out there that we haven't really yet explored. I think the first generation of Unnatural Products, the first technologies we built were all about, can we take a binder someone else has found or a binder that already exists and optimize it to have, the cell permeability, solubility, ADME properties of something like cyclosporin while maintaining its potent binding. And that was a little limiting because we were, had to go to a pharma partner who had an active series and collaborate with them or pick molecules up from patents or public domain. We've just now brought on some of these de novo discovery technologies internally. So I think we finally have the ability to pick a protein target and really go from biological hypothesis to molecule and then optimize that into a drug-like lead. So I think I'm really excited to see some of those new protein targets that we don't even have tool molecules for kind of yield to these peptide-based approaches. And I think that's where you can get really surprised and find interesting biology when you're developing molecules against protein targets and pathways that have really been underserved. One thing that I always think is a little funny is the Macrocycles elucidating biology goes all the way back to the beginning, like cyclosporin, rapamycin, and FK506. the proteins are named after them, not the other way around, right? Cyclophilin binds cyclosporin, FKBP binds FK506, and mTOR is the target of rapamycin. So I think I'm excited to see, these new Briand Rule of 5 compounds continue to elucidate new biology, and then ultimately, help treat disease in new ways.
Nice. Cam, was there anything that you wanted to add?
No, I mean, I think, like Josh said, I think we really were in the salt mines for the first part of the company. The tech we were doing was building, was really rooted in solving this medchem challenge. And now, our onboarding all these de novo discovery technologies and putting them to practice over the next couple of years is just going to be super, super exciting at UMP, because we know those work, right? We know those can generate ligands against challenging targets. It's always just been about what comes next. And now we have that piece of the puzzle too. So I think we're already starting to see it, with early proof of concept of some of those programs just starting to bear really exciting fruit internally. I think the next 18, 24 months are going to be pretty exciting times here at Unnatural Products. And then, I think just like you underlined earlier, just the field at large. I mean, I think that the modality landscape is just really growing. There's public markets have been doing some unfortunate things in the biotech sector lately, but there's been a ton of money poured into the private sector of a lot of interesting and innovative new technologies getting booted up in the biotech space with I think a tolerance that really hasn't been there before. And I think not all of those are going to be winners, but I think there are going to be some really impactful new medicines that come out of this kind of current crop of new really hyper innovative platform technologies and biology companies that are being generated. And we hope to be one of them, but I certainly, with the honor to be a peer amongst a lot of other really exciting modalities that I think are really going to transform medicine over the next 10, 20 years and what's possible. So I think I'm excited to be a part of it and looking forward to learning from others along the way.
Yes, awesome. Is there anything that maybe I didn't ask about that you would like to, let people know about Unnatural Products?
You didn't ask how many people are trying to hire in this beautiful location of science campus.
Talk about your location.
So you can see behind Josh there. We're based in Santa Cruz. I'm just up the road. Still gonna dog walk on my agenda this morning. I think we've been growing like crazy. We just are moving into 4 brand new fume hoods that we just built out, expanding our kid lab this week. And I think the plan is to continue growing from here. So I think definitely if there's peptide chemists, medicinal chemists, pharmacologists, people that think about ADME a lot and beyond Rule of 5 compounds that listen to these podcasts, look us up. We'd love to work together and be part of that kind of grand vision I just said earlier. So,
excellent. All right, great. Well, thank you both for your time. I appreciate it. If there's any resources that you would like to send me links for, I'll put that in the description. Awesome. And just thanks again.
Well, thanks so much for the interview and the time, Wendy. It's great to see it in 2 dimensions and look forward to doing it in 3 dimensions soon.
Yeah, thanks a lot, Wendy. This is super fun.
Yes, absolutely. Thank you for tuning in to this episode of Exploration Science. If you enjoyed this content, please like and share with your communities. As always, we appreciate your feedback as well as your suggestions for topics that you would like to see covered. Thanks again for tuning in.